Thank you very much for that kind introduction, and I'm very pleased to have been asked to speak this morning, particularly with Dr. Bartha Knotters following me. Newfoundland and Labrador has been a very exciting place to do genetic research. And this is really because of the original settlement in patterns in Newfoundland and the large families until very recently. Because of this, there are clusters of what are otherwise rare genetic diseases, and initially these need diagnosis and treatment, but they're also very valuable for genetic research. Both for understanding the clinical features of these conditions, but also help in identifying a gene and mutation. So this can be helpful initially to the families, but then much more broadly. The settlement of Newfoundland was primarily initially in the late 1700s and early 1800s from southwest England and the Devon, Dorset, Hampshire area, and southern southeast Ireland in the areas of Cork and Waterford. Oh, okay. So there had been information from early explorers that there were very rich fish stocks off Newfoundland, and that was really the reason for the immigration. So small groups of 30 to 50 would travel in a ship like this to a coastal area of Newfoundland. They would settle, for instance, a group from Hampshire in Greens Pond, Newfoundland, another ship from another community in another area along the coastline. These are called outports, and even the outports that were geographically relatively close together as a very rugged coastline, they remained very isolated for long periods of time. So if you had another group from another area settling in a separate area, they're completely separate from each other. We know that everyone has some mutations. In a large population, there might be a few people with any particular mutation, but if there's a cluster of people who are moving to the New World, they may very well be related, and so there might be several I'll get this coordinated shortly. In that group with a particular mutation, when this group then moves to the New World, that mutation will become much more common in that particular cluster. It then becomes likely over the generations that any couple might be both carriers of the same recessive disease, and then there's a risk of that condition. What we see around Newfoundland is many different clusters of rare disease. So in each group that came, there might be a different mutation. My past was Drosophila genetics in Newfoundland in the 1960s, moving to Newfoundland in the early 70s. My first opportunity to work in Newfoundland was in 1978 when I met an ophthalmologist who was very interested in hereditary eye diseases. With the help of a small grant from CNIB, we started a weekly ocular genetics clinic, first in St. John's, but then as we realized that a number of the patients we saw had relatives in their rural communities, we began having outreach clinics in the rural hospitals. I also reviewed the CNIB records. These were paper records at the time. They had no way of determining the prevalence and distribution of hereditary eye disease, and so I read all the records. There was tremendously interesting information that was very helpful to identify clusters of disease. And this particular information was from a family, a region down here that is now resettled. And even at the time I read the information in the 80s, many of these communities had resettled, but there was information from the CNIB field workers from the 20s and 30s in some of these people. And so I was able to document many hereditary eye diseases around the province. When this was all reviewed, if there were individuals who seemed to have clusters of genetic disease, but a specific diagnosis wasn't known, we would then invite them to be seen in one of our clinics. And when it was all reviewed, there were 29.5% of the just over 1,000 registered blind who had definite hereditary eye diseases. There was probably some others who were also in some of those groups. These were all subdivided into specific diagnoses. The most common were the RP and retinal dystrophy. These are the rod and cone disorders, RP with night blindness and peripheral vision loss initially. There's the this group here in the blue. We've done all of the clinical and molecular studies. Some of these others are underway, but our attention now is particularly on these most common retinal dystrophies. But what you see again is the many different families with the different conditions and different families with RP around the province and so on. One particular group that was much more common in Newfoundland, Bardet-Biedl syndrome, a complex disorder, was 10 times as frequent in Newfoundland as described worldwide. There's very early blindness with possibly complete loss of vision in the late teens, but there's also kidney and endocrine abnormalities, possibly polydactyly, and the old literature all said they were mentally retarded. Because of this, in Newfoundland and in other places, the label meant that many of them were not given a chance for schooling. One of the things we did in the 1980s was a clinical review of all of the features of the patients in our families. And one of the most important things was that we got an IQ test that was designed for those with severe visual impairment. We were able to show that less than 1/3 were in the mentally retarded range. Others had developmental delay, but everyone then was able to have the schooling, and at that time there was schooling designed for those with visual impairment. Some even completed university. There also was much more attention paid to the other medical problems that these patients had, the early diabetes, early hypertension, the kidney problems. We and others had thought that there would be, initially had thought that there would be one founder mutation for BBS in Newfoundland. But the mapping of genes actually showed several loci in different families. And then the first gene that was actually identified, the BBS6 gene, was through our study of this particular Newfoundland family. No one had any idea what the function or location in the cell for the genes for BBS might be. But this gene was very helpful in leading to this. We now know 9 different mutations in 6 different genes, so a very diverse group of mutations in these families. But the first BBS6 gene was shown to be related to cilia structure and function. And the cilia at the time was thought to be rather a vestigial structure with very little importance. Because of the location of the first BBS gene to the cilia complex, that was the location to look for other genes for other BBS families. And there now are 17 or 18 BBS genes in total. There is this tremendous complex of genes that are BBS, but a group of other related disorders with overlapping features, and these are all now referred to as the ciliopathies. They tend to have decreased vision, there's endocrine and kidney abnormalities, there's brain malformations, polydactyly, and so on. And when I was looking for these diagrams, I found the picture at the top on the web, and that was when we were looking at the pedigree of the first BBS family to be mapped. Then one day in 1982, there was a new direction. There was a referral to the eye genetics clinic which said, eye, brain, and kidney tumors, please see. Several sisters came to the clinic And they were telling us about their father dying at 41 of brain and spinal cord tumors. There were other early deaths in relatives as young as 18 and 21. They couldn't tell me exactly what. Fortunately, Gordon Johnson was able to recognize this vascular cluster on the back of the eye as a retinal angioma, and this is a feature of von Hippel-Lindau disease. Other manifestations in VHL are also vascular tumors in the cerebellum, the kidney, the adrenal gland. And so what the family called this condition was the curse of the lanes. They didn't know why, and nor did the doctors know why. There were so many early deaths. There was blindness, there was brain tumors, neurological deficits, and sudden deaths. So the first thing after the clinic was that I went to the library. I had never heard of VHL before. The second thing was to talk to the family members shown here that people could put me in touch with to get consent to review all of their records and to determine exactly what type of tumors they had and at what age. And the third thing was to bring together a group of physicians, and this was with the help of Ian Bomer. So this was including pediatrics, internal medicine, radiology, neurosurgery, nephrology, and so on, as well as our genetics and ophthalmology input. And to assess the types of tumors that were seen, what age had they been seen, what did the literature say, and there was very little in the literature about screening at the time. And so we set our screening investigations. A test to look for each of the manifestations that we were aware of. We started at early teens, but some of the individuals who were asymptomatic came in, they had a retinal angioma or an adrenal tumor at their first screening. So we continually reviewed the screening and moved it to earlier ages. We added imaging, initially CT scan when it became available, and subsequently we've switched that to MRI to avoid the radiation. There had been 25 or 15 individuals that we could show that were affected initially. Within just over 10 years, there were 30 more shown to be affected, but there were also at least 100 other tumors that were identified in individuals who were asymptomatic. There had only been 3 deaths in that time, but they were people who had been symptomatic prior to our recognition of the family. So this was a huge family with a very serious disease, many people with many different manifestations. This individual here with the 4 sectors filled in and the star meant the 5 types of tumors, and she has had at least 8 different renal cell carcinomas, all now treated successfully. This family was part of the linkage of the VHL gene, and a mutation was found in this family and 5 smaller families that we had identified during the follow-up of this family. We've been monitoring the types of tumors ever since, and the one disconcerting feature is that pancreatic tumors, which had not been seen originally, were becoming increasingly common. They're malignant tumors, pancreatic neuroendocrine tumors. They're difficult to see, although radiology is revising methods. They're also difficult to treat because they're diffuse throughout the pancreas. So the screening protocol is much earlier, and as I said, MRI rather than CT scan. Then there was a referral from a family doctor on New World Island. Dr. Sheldon recognized that several patients who had symptomatic colon cancer had multiple polyps in the pathology. He knew they were related. He asked for help with determining who else might be affected or at risk. And over a 2-year period, I interviewed people, and there in fact were 60 affected and many others at risk. The pathology reports were looked at in detail because, particularly to do with earlier generations, people might say, I think Aunt Mary had the same thing. It might have been the same thing, but it might have been surgery for ulcer disease or an ovarian cyst, and so this was very important. it turned out to be very helpful because this family was very different. In typical FAP, there were hundreds or thousands of polyps throughout the colon, increasing in numbers towards the right colon, and so a sigmoidoscopy was the screening recommendation. In this family, many people had only 10 or 20 polyps. They hadn't been recognized as being affected. And a third of the family members had polyps only in the right colon. So a sigmoidoscopy would be normal, while they might be developing a cancer in the cecum. So for this family, it was necessary to recommend colonoscopy for screening, and this is then called attenuated or atypical FAP. Information was given to the family members, but also to the family doctors and to the surgeons who had been doing the scopes. Interestingly, they then identified some other families with similar clinical features, and also from central Newfoundland. We were able then to identify a common ancestral couple of all of these 5 families, and later a common mutation. So these families all have the same genetic test, the same clinical screening. This led then to a referral of another family with FAP, one of the family doctors who'd seen a branch of that family. At the same time, one of the surgeons said, there's a man upstairs who says there's too much colon cancer in his family, and please see him. This was in about 1990. And so the first discussion with these families, initially this young woman who was 43, she'd had 2 colon cancers, but she'd also had an endometrial cancer. In the second family, one sister had had 2 colon cancers, the other had had endometrial cancer. I was introduced to other family members. There were frequent endometrial cancers in the women, as well as or instead of the colon cancer, and this is not part of FAP. Also, the patients did not have multiple polyps. So this was a condition then called hereditary non-polyposis colon cancer. I did the pedigree studies, the medical record review, then archival searches to take the pedigrees back as far as possible. The 2 families both came from the northeast coast, about 200 miles apart, but we knew that people had moved westward from that Wesleyville, Greens Pond area. In the early 1990s, the FAP gene had just been identified. The same labs were interested in finding a gene for HNPCC, but they didn't have any family large enough. You needed a family with many DNA samples from affected and unaffected family members. There were many families that were dramatic on paper, but mostly it was following the death of affected individuals. So in 2 years, I actually had then 2 pedigrees. It was this part, but it included this early generation. And this part here, up to this woman here, but no one could tell me her maiden name, so this was not joined at the time. Then one very fortunate day in Fortune Harbor, Newfoundland, I went to see the last person here. She didn't know any further family history because she was very young when her father died at 29. And finally she said, well, I must give you a cup of tea before you drive home. She called in her husband, who was a fisherman. He was very uncomfortable with it, come from away, and he brought the family Bible. There was nothing inside the front cover. There was a piece of paper inside the back cover. It was the marriage certificate of this person here. She had the same name. She came from the same community as the other family. Her parents' names were there. They matched perfectly. So this was a large family. And this was the family then studied by Bolstein and de la Chapelle in trying to map the first HNPCC gene. It was 1 marker at the time. It was a 346 marker, but the location and then immediately the gene MSH2 was identified. What you also may be able to see is under each of these affected individuals, there's 1 or 2 or 3 lines of information, and those are the tumors they had, the types of tumor, and the age that they had them. So this gave us then the types of cancers that were seen more commonly in HNPCC than in the general population. These types of cancers and the age that they occurred then allowed us to set screening recommendations. And so as I was seeing all of these family members, screening recommendations were given, but there were revisions provided to families and their family doctors as we knew more information. These transitional cell cancers actually occurred in one specific group of cancers. One thing also that was fortunate, other molecular geneticists remembered their yeast genetics. So the gene PMS2, which is here, was known in yeast genetics to be part of a cluster of genes that worked to correct mistakes in DNA. When the cell divides, the DNA must copy first. There's some mistakes. There was the mismatch repair system in yeast to correct those mistakes. There was also this system in humans, and surprisingly, it was the basis of this human disease if there's a mutation in any of those genes. So HNPCC families that didn't have MSH2 mutations often had mutations in these other mismatch repair genes. So again, a new mechanism for cancer predisposition in humans. Many other families were referred as we were sending information back to family doctors and surgeons, and even initially there were 12 families that we tested for the mutation, the family C mutation as it was found, 12 families who lived across the province but could all say they came from Bonavista North. So now we know 17 families with that mutation. There was another group of families that came from a slightly different area, and they were the ones with the transitional cell cancers. They didn't have the family C mutation, but they all had a second mutation in MSH2, the deletion exon 8, and so they have a slightly different screening protocol because of the extra type of cancers. So there are many being screened. There was a much better understanding of the disease in the families and the family members, and so much less stressful for them. There was the early identification and treatment, so fewer cancers and fewer cancer deaths. But in all of the HNPCC families, besides these 2 founder mutations, we found 12 individual mutations, 1 per family, but many families with HNPCC with no mutation, and that's the same elsewhere. So there's other genes unknown. Now, back to the eye diseases. This was being carried on while all of the hereditary cancer work was being done. But by the late 1990s, many of the genes for some of the hereditary eye diseases, the retinal dystrophies in particular, were being identified. It's very important to identify those with the hereditary eye diseases because of the implications for education and therefore for employment and for family life. These are conditions that typically present in childhood or the teens, and so they have a great impact. But also by mid-2000s, the treatments were being developed for some specific hereditary eye diseases. You had to know the gene that was defective before you could be available for treatment. The seeding process is extremely complicated. Every one of these genes could be involved. So a mutation in one gene could cause a very similar clinical disease to a mutation in another gene. But if there was a treatment being developed for mutations in this gene, it was only for this gene. So the first type of treatment was gene therapy, where the gene was attached to an adeno-associated virus which had been inactivated. There was injection under the retina to be picked up by the photoreceptors. And we hope that some members of the families in Newfoundland would be able to avail of treatments in the future. So there's many RP families with recessive inheritance, with dominant inheritance, one with X-linked inheritance, and many families here where there's just one affected, so we don't know the inheritance pattern. One very successful identification of a mutation was there was 3 small families. Each of these individuals were seen in different locations, but they all said they came from the Northern Peninsula. Through genealogical studies that we have now, we were able to show that they were related to each other. It's always helpful to have many relatives affected and DNA from affected and unaffected to map a gene. Through association with FORGE, the clinical and molecular group across Canada, we were able to find a mutation in this gene MERTK, and we expect many more carriers in that community and probably others with RP. But MERTK is one of the genes where people in Florida are working on a treatment. Stargardt disease is hereditary juvenile macular degeneration, the same type of severe central vision loss as in age-related macular degeneration, but starting in childhood. So these are our families. One gene, a very large gene, the mutations primarily known, a cluster in Conception Bay, but all told there's 17 mutations in Newfoundland families and 15 occur in that one cluster. So through all of the genetic studies in Newfoundland, we see different population-specific frequencies of genetic disorders. So there's different healthcare needs throughout the province. This is important for diagnosis, for the screening and treatment, but also for the education of patients and families. And this is one thing that we've been involved in right from the beginning, with letters and guidelines, presentations, now telemedicine sessions. I visited many of the rural hospitals to give talks to the physicians about the conditions in their area, but also to families in the area. Talk to undergraduate and postgraduate medical students and subspecialty groups, and also to the professional organizations, because many Newfoundlanders have moved to other parts of Canada, but the theoretical part of the work that's being done in Newfoundland can apply in other places. So just as Lap Chi Choi said yesterday, serendipity has had a lot to do with my career. Thank you very much.
Thank you, Jen. I think we have time for perhaps 1 or 2 brief questions. Dr. Hachinsky?
Fascinating work. Congratulations. Did you provide genetic counseling? Did that make a difference as to whom members of the affected families married?
In most cases, in the hereditary cancers, these are dominant conditions, and so it was really a question of whether their children might be affected. And all through the 80s and 90s, I did all of the genetic counseling with all of the hereditary cancer families as the research was being conducted. It did make a big difference about whether some of those who were recognized to be affected had children or not. They didn't want their children to have the same kind of disease that they had or others in their family had, and so that was definitely a difference. With some of the hereditary eye diseases, we tried to give them the information and particularly help with identification or examination of children of people who might be carriers. So if there's the same eye disease, to find it and have the proper schooling early. These conditions, the recessive conditions, they're not so likely to make a difference in whether people have children, but it's to make the diagnosis early and manage accordingly, so that this is all provided to family members.
And one more question, Dr. Ling.
Thank you for a great lecture, Jane. I'm wondering, is there an opportunity to investigate people with mutation but without the disease? Because that could give a huge clue that to the suppression mechanism, whatever that
Yes, I mean, we've seen that because in the FAP family, there's a tremendous variation in the age of onset. The youngest, there's some 10-year-olds who've required colectomy because of the multiple polyps. When they see the multiple polyps, their colon is removed to prevent the onset of colon cancer. Some others in the same family, same mutation, have not had any polyps until 40, even though they've been screened. So obviously there was a difference. In the large HMPCC family, there was one person who was 80. She had affected daughters, a 27-year-old grandson who was affected. When the mutation was known, she clearly had it. At 82, she was willing to go for a colonoscopy. The colon was clear, and she died shortly after of heart disease. So what is the difference? There's not enough people in the different families, really, but exactly what you're saying. If we knew why she had no disease so late or others had disease at 60 instead of 20, that gene that they have that's protective might be very valuable to other people. So it's something that needs to be done.